Hi, today we're going to learn about DNA replication. The self-complementary nature of DNA allows each of the two strands of a parental DNA molecule to serve as a template for the formation of a daughter strand. This process is called DNA replication. However, replication of even the simplest DNA molecule is a complex multi-step process involving several enzymes. When you have completed this exercise, you should learn or understand the difference between the leading and the lagging strand, understand the functions of DNA replication proteins, and understand how the DNA polymerase is able to copy two anti-parallel strands simultaneously. Many proteins are involved in replications. These proteins include DNA helicase, single-stranded binding proteins, topoisomerases, primase, DNA ligase, DNA polymerase, sliding DNA clamps, RNase H, and DNA ligase. DNA helicases are a class of enzymes that couple ATP hydrolysis to the separation of DNA strands. The DNA helicases involved in replication are typically hexameric proteins in the shape of a ring. The junction between the newly separated template strand and the unreplicated double-stranded DNA is called the replication fork. Replication fork can be looked at as a moving continuously towards the unreplicated double-stranded DNA. Single-stranded binding proteins, or SSBs, bind to the single-stranded DNA to stabilize the separated strands. As the DNA unwinds, the twist number decreases, therefore the right number must increase, causing the DNA strands to become positively supercoiled. These supercoils must be removed by topoisomerases. DNA requires, or replication requires, an RNA primer to begin. Primase is a specialized RNA polymerase which makes short RNA primers using single-stranded DNA as a template. Primase is activated by the association with DNA replication proteins such as DNA helicase. The synthesis of DNA is catalyzed by an enzyme called DNA polymerase. DNA polymerase is able to add new nucleotides only to the 3' end of the polynucleotide strand. Due to the anti-parallel nature of the DNAs, one strand is synthesized continuously towards the replication fork and the other strand is synthesized discontinuously away from the replication fork. The one towards the replication fork is called as the leading strand, and the one away from the replication fork is called as the lagging strand. The continuously synthesized strand is called the leading strand, and the discontinuously synthesized strand is called as the lagging strand. It takes approximately about 1 second for the DNA polymerase to bind to the DNA. However, once replication has begun, DNA polymerases are capable of adding about 1,000 nucleotides to the growing daughter strand every second. The ability of an enzyme to catalyze many reactions before releasing the substrate is called as processivity. To increase the processivity of DNA polymerase, a sliding clamp or the clamp surrounds the DNA and binds to the DNA polymerase, holding them together. Remember, DNA polymerase can only add DNTPs to the 3' end of a polynucleotide. Because both DNA strands have to be passed through the replication machinery in the same overall direction, the lagging strand is synthesized discontinuously in small fragments. The short fragments of DNA formed on the lagging strand is called as the Okazaki fragments. Note, although the Okazaki fragments are shown as very short stretches in this animation, they are usually about 100 to 1000 base pairs in length. To complete DNA replication, the RNA primers must be removed. RNase H specifically degrades RNA that is base paired with the DNA, and the H stands for hybrid, as in RNA-DNA hybrid. The single ribonucleotide directly linked to the DNA is removed by a separate exonuclease. Removal of RNA primers leave the single-stranded gaps in the double-stranded DNA. These gaps are filled by DNA polymerase. Finally, the nick between the 3' hydroxyl of the repaired section and the 5' phosphoryl of the replicated section is repaired by DNA ligase. In bacteria, the two enzymes responsible for replicating the leading and the lagging strand are linked together by a multi-protein complex called holoenzyme. The trombone model explains how the replication machinery might exploit the flexibility of DNA to accomplish this task. According to this model, a single-stranded DNA template for the lagging strand pulls through the DNA polymerase, similar to the sliding of a trombone. This flexibility allows the DNA polymerase to add nucleotides to the 3' end of the growing lagging strand. The DNA polymerase holoenzyme also contains a sliding clamp loader. This multi-protein complex loads sliding clamps onto the primed DNA. Sliding clamps help the DNA polymerase find the primed DNA and then help it to increase the processivity of the DNA polymerase. Hope you understood DNA replication. Thank you.